All right. Welcome, everybody. We've made it to, to day 4 of a workshop, and you have learned so many tools. And I just want to give you a little glimpse of beautiful University of Georgia in the spring. It's absolutely gorgeous here, and hopefully workshops will be returning to in-person sooner rather than later. So today's module ties together so many of the tools that we have learned over the last several days. You'll have a lot of fun with it. You might even want to revise some of those final projects. We'll see. All right. And so ViewPathDB maps gene products, if it's an enzyme and has an EC number, to pathways using that EC number. And we talked about EC numbers in one of our earlier exercises. These are enzyme commission numbers. Right? And so we import pathways from a number of sources, primarily KEGG, but also Metapsych in some instances. And we can map these gene products onto those pathways. But we have developed within our framework the multiple ways to interact with these pathways and include them in your strategies and conduct a number of analyses. So our exercises today will play around with that a bit. Interestingly, just as we could take genes from one organism and transform them into genes in another organism, we can take metabolic compounds and transform them into the pathways that act upon those metabolites or the genes that are present in those pathways. So we have another type of transform that you'll be introduced today. We can take pathways and we can do really useful things. We can paint them with orthology information. So for example, imagine looking at pathways and comparing whether or not the ortholog is present in the pathogen and the host or across all the species in your genus. This becomes really useful if you're thinking about trying to find a therapeutic target or understand conservation of a biochemical pathway or process. We can also map onto pathways data from transcriptomic, EQ2L, or proteomics experiments. Today, we'll do a transcriptomics exercise where you can actually look how gene expression changes throughout whatever the experimental conditions are relative to the enzymes that participate in a pathway. Can also locate, you remember earlier, we looked a lot about cell location. And so it's important to think about where these pathways and processes happen because they can happen in different compartments in the parasite. And we can also examine how compounds or metabolites that are acted upon in these pathways change during experimental conditions by studying metabolites. And so much like we've used tools to look at how gene expression changes under experimental conditions. We will play around with an exercise today that looks at how metabolites can change during experimental conditions, right? So sort of where we're headed. So to get started, this is a Plasmodium metabolic map. It doesn't include everything that's going on in the parasite, but it handles the main points that I'd like to talk about today. One is to think about space, right? And this isn't a part of our exercises, but I think it's important to think about which biochemical processes are happening where. What is happening in the mitochondrion or in a food vacuole or in a plastid or in the cytosol? And where do metabolites come from? This row up here represents transporters, and the parasite is interacting with its environment. And as it is a parasite, very often it's lost the ability to produce certain compounds and has to obtain them from the host. Some pathways are shared between compartments. It gets pretty wild. That's why parasites and protists are so cool, as are many of the other amazing organisms that we work on in our resources. I mean, it's just exciting biology. So often we think about looking at metabolic pathways with respect to drug development. And so I think it's important to realize that we can start by looking at a pathway if there's already a target that's known. I'll give an example in a minute of kinases. Kinases are very popular drug targets. So it'd be really nice to then see whether or not that target is present in your pathogen, or is it present in the pathogen and not in the host, et cetera. So that's sort of a rational drug design approach. But it's also used extensively for drug target validation. In other words, if we don't know the mechanism of action from a chemical screening of compounds and you're trying to figure out what it's doing, you often like to look at changes in gene expression, changes in the metabolites, in response to, if you think you now know the target, you might knock it out and ask, is that really the target, and look at what happens with the phenotype. And so there are many reasons to have a look at the metabolic pathways, including studies of resistance. So very often people look at pathways and they're thinking about drug target design, which is not the only reason to look at pathways, because they're looking for choke points, right? They're looking for bottlenecks in which the pathogen or you know, where you can attack, where the parasite may not have, or the fungus, whatever our pathogen is, or the mosquito may not have an alternative way to do something. And I just want to point out that just because on the metabolic map it looks like a choke point, in other words, if we knock this out, we couldn't get anywhere, but organisms are incredibly resourceful, and they might pull in a metabolite from the host and feed it into that reaction, or take another reaction intermediate and pass it on to another enzyme. And so very often something looks really good, and success really depends on you know, that final experimental validation. So this was a nice example tying together some lectures this week. So we may remember talking about HXGPRT when we were looking at gene structures. And this enzyme is present in most AP complexins as part of a purine salvage pathway. But in Cryptosporidium, HXGPRT is missing. All right, and it looked like this enzyme right here, IMPDH, might be a good target as a choke point reaction, because if the parasite cannot synthesize, right, guanine, it's really in trouble. And so it was really neat too to realize that that particular enzyme, IMPDH, when you looked at it in terms of orthology, it turns out that it's a horizontal gene transfer. In other words, the enzyme in Cryptosporidium is most closely related to a bacterial enzyme, which is great because it gives you discrimination between host and pathogen for a drug. And it turns out that IMPDH is a known drug target. And so there are already compounds that could be tested for repurposing as a possible anti-parasitic in this case. And it's really nice to see that when you take these compounds that are still undergoing medicinal chemistry modifications, but they actually did have an effect on parasite growth. And so very important to many of the areas that folks like to study. I want to also return to our discussion of paralogs and orthologs, because many proteins that are important to the organisms that we study, in this case, I'm showing you an example of kinases, do exist as family members. And so it's important to remember that there might be more than one copy of a gene. And while that doesn't happen as often for some of the enzymes, it does for others. And kinases, in this case, it happens tremendously. So this sort of bar graph here is showing just a rough schematic of relationships within the AB complexa. And I'll just point out, for example, that in the coccidia, this group of kinases called the ROP kinases has really gone to town in terms of its number. So these are the number of kinase domains identified in the genomes. And you can see that there's tremendous variation between the organisms and sort of their gene content and the distribution of the types of family members. And that's when it becomes really important when we're doing those orthology analyses to pull up and make multiple sequence alignments of the types that we did in the exercises, where you can actually begin to look at and get an idea about which amino acids may end up actually proving to be involved in the active site and or changing the function of that active site if they were interfered with as a target. So I just give this as an example. All right, so the enzymes, which are proteins, are encoded by genes that act on chemical compounds, right, metabolites. And so just like we can use transcriptomics to study gene expression, we can look at metabolomics to understand what's going on in pathways. And so I think it's really important to also explore not only the pathway and the enzymes in the pathways, but realize the power of the tools that are available to actually look at what's going on inside the cells. And we have several metabolomics experiments, and we're going to look at one today. But it's really powerful. And much like we look at gene expression studies, you have different conditions, right? And under condition 1 and condition 2, you have your reference and you have your comparator. And you're going to now, instead of isolating RNA transcripts, you isolate the metabolites, you run them through mass specs, you have ways of labeling and keeping your reference and your comparator separate from each other. You go in, you look at mass spec profiles in the end, and you end up by identifying metabolites that might be different between your conditions, or levels of metabolites that are different between your conditions. And it's a very nice way of trying to get at the phenotype of what might be going on in your system. And it's a really valuable data set that can be integrated. All right, so In the interest of time, I've given you lots of additional resources, places where you can go for looking at known drug targets or looking at various drug target databases, small metabolite databases, transporter databases, OrthoMCL, ViewPathDB, et cetera, so that you can begin to think of some of the additional resources you have that might be useful to your research. In this module today, we're going to learn about finding and exploring pathways. You're going to learn to visualize experimental gene expression data and orthology data in the context of a metabolic map. And on those maps, I just want to let you know that you'll see in the exercise, some pathways are very large, so you might need to zoom in. And sometimes things can be a little confusing. You can actually move them around a bit. It's a neat tool to play with. And each of the nodes, the points connecting the lines on the pathways, will have information available to tell you more about what it is that you're looking at, either the enzyme or the metabolite or the experiment that's mapped to that pathway. You're going to learn how to mine specific metabolic compounds to find a particular compound in that malaria pathway I showed you today. The enzyme right in the middle of that slide was PEP. We're going to, or I should say the metabolite was PEP was right there, and we're going to look at that. You're going to learn how to mine metabolomics data, and you'll notice that the tools are very similar to transcriptomics data. And you're going to learn how to transform the compounds that you find from mining metabolomics data into the pathways, or if you choose the genes, encoding the enzymes that act upon them. And so with that, I would like to wrap this up. I don't know if any of the other instructors wanted to contribute something, or if we just want to head into our exercises, our last ones.
I think we're probably good to go to the exercises. Just checking to see if there are any questions. It doesn't look like it. Any other comments? Okay, I think Rachel, you can send us off to the breakouts.
Yay. Bye, everybody.